Dear audience, I would like to start with a scientific discovery made by two German scientists more than a century ago. So they took an animal and they injected a blue dye into its bloodstream. And what they found is that the whole animal was stained blue except for one part, the central nervous system. Later, this experiment was repeated the opposite way, so that they injected the dye into the brain of the animal. And now nothing but the brain was stained blue. So this confirmed the hypothesis that there is some kind of barrier that separates the circulatory system and the brain. We now call this the blood-brain barrier, or shortly, BBB. The blood-brain barrier is a term for many specific features of brain capillaries, the smallest blood vessels that are found in your brains. These brain capillaries are made of cells called endothelial cells that are really specific in the brain because they are in constant communication with two other cell types called pericytes, or neighboring cells, here in yellow, and astrocytes, which are star-shaped glial cells, here in blue. To learn more about why these biological barriers are important, especially the blood-brain barrier, and how we can model them in the lab, please check out Professor Maria Deli's lecture this afternoon. What I would like to talk to you about today is a biomedical problem associated with the BBB. Because the problem is that it not only restricts the brain penetration of blue dyes, but also most of therapeutic drugs. And therefore, brain diseases such as Alzheimer's disease, Parkinson's disease, epilepsy, or brain tumors are especially hard to treat pharmaceutically. So how can we solve the problem? Well, there are many approaches for this problem. I will just focus on the one that we also use in our lab, and that is using nanoparticles as molecular Trojan horses. So the idea is that we can load the drugs into nanoparticles, particles that are 10,000 times smaller than a millimeter. So we load the drugs into them, and we can decorate the surface of these nanoparticles with targeting molecules that are specifically recognized by the cells of the BBB. Something like this. And a new promising class of candidates for this targeting are nutrient transporters found in brain endothelial cells. So because the brain has a really high metabolic need, these proteins come in large numbers in these cells, and yet they are not fully exploited for drug delivery purposes. So we selected two targeting molecules, alanine, which is an amino acid, and glutathione, which is a tripeptide and was previously shown to be effective. And we hypothesized that if we label our nanoparticles with these two targeting molecules, or specifically their combination, there would be an increased uptake of the nanoparticle cargo in brain endothelial cells and an increased penetration of cargo, so how well they can go through the BBB in this BBB model we made in the lab. But more on that in the afternoon talk. So we prepared 4 types of nanoparticles called niosomes from non-ionic surfactants and cholesterol. 1 untargeted niosome and 3 targeted niosomes that were labeled either with alanine, glutathione, or their combination. We chose a high molecular weight fluorescent protein complex called Evans blue albumin, or shortly EBA, as the cargo of the nanoparticles. We made the nanoparticles using a method called lipid film hydration that was followed by a high-pressure extrudation step through a polycarbonate filter of 100 nanometer size. We then used ultracentrifugation to separate the unencapsulated cargo from the cargo that was successfully encapsulated in the nanoparticles. We also characterized our nanoparticles by their size and their surface charge. As you can see, the diameter of niosomes was around 100 nanometer, and they had a slightly negative surface charge. So before moving on into any further experiments, we had to test whether our nanoparticles were toxic to cells and determine a safe concentration to use. So for that, we used a real-time impedance-based cell analysis. Basically, we treated the cells with niasomes in a wide concentration range, and after 4 hours of incubation, we found that none of these treatments had any significant damage to endothelial cells, so therefore, we selected the 10 milligrams per ml concentration to work with. Then we examined the uptake of this fluorescent cargo into brain endothelial cells. So we incubated the cells with niasomes for 4 hours, then washed the cells, lysed the cells, and then quantified the fluorescence intensity of our cargo by fluorescent spectrophotometry. Compared to the non-targeted group, the cellular uptake of unencapsulated, so free ABA, free dye, was very low, around 1.5%. The presence of targeting molecules on the surface of niasomes elevated the cellular uptake of the cargo, especially dual ligand labeling. We also confirmed this by confocal microscopy. Here you can see the nuclei of the cells stained in blue and the red fluorescent signal of ABA. So again, this is the free dye. This is dye that was encapsulated in non-targeted niasomes, and this is the dye that was encapsulated in dual targeting niasomes. I think you can see the difference. And finally, we were curious about the permeability of the cargo across the blood-brain barrier, so again, the penetration across our BBB model. So we treated the upper compartments with niasomes, and then after 4 hours of incubation, we collected the lower compartments and again quantified our results by fluorescent spectrophotometry. Again, compared to the non-targeted group, the penetration of unencapsulated free ABA was very low, which reflects a good tightness of our model. And again, labeling niasomes with either alanine, glutathione, and especially their combination resulted in an elevated BBB permeability, dual ligand labeling being the most effective, producing an 8-fold increase. So to sum it up, our research group is focusing on developing a new drug delivery system for brain drug delivery. For that, we use nanoparticles that are targeted with either alanine, glutathione, or their combination to target nutrient transporters in brain endothelial cells. We showed that these nanoparticles did not damage the endothelial cells, and the labeling of these nanoparticles with targeting ligands increased the cellular uptake and the permeability of the cargo in our culture system. So we conclude that targeting nutrient transporters at the BBB could potentially be exploited for drug delivery to the brain. With that, I would like to thank all of the members of the Biological Barriers Research Group in the BRC Szeged, especially my mentors, Professor Mária Deli, Dr. Szilvia Veszelka, and Mária Mészáros. You can also check out our new Facebook page under the name Biológiai Barrierek Kutatócsoport. I would also like to thank the Szeged Scientist Academy for supporting me, and thank you for your attention.